All right, we're going to begin into the nervous system, and we'll start with chapter 12. This is going to be looking a lot at the microanatomy of the nervous system before we get into some of the larger structures and the physiology of the nervous system. This is kind of the last section in what we will be learning for this course because the next several chapters all have to do with different aspects of the nervous system. So to start, this should not look unfamiliar to you because in the beginning, in chapter 1, we learned about homeostasis, where we are able to collect information, send it to the brain, and then have some sort of reaction. And we learned those reactions as negative or positive feedback. Okay, so in general, especially in regards to homeostasis, this is how the nervous system functions. First, we collect information. We have some sort of stimulus. This is a change that occurs in the body, and it is detected by receptors. Okay, and there's a lot of different names for receptors that we'll get into for the nervous system. Those receptors, once they detect a change, are going to send a signal to the control center. And the control center is going to be your brain or your spinal cord, okay, for the nervous system. And they are going to basically compare this change to whatever set point was set inside the body. So they process the information for whatever the change that was occurring, and then they'll send it a signal to an effector. And an effector is always a muscle or a gland, and that effector will have a response. I can give an example of this, okay, and we'll have many examples as we go through here. So say your hand touches a hot stove, okay? The stimulus is the heat from the stove, and the receptors, okay, are in my hand. They could be temperature receptors, which are called thermoreceptors, or they could be pain receptors, like nociceptors. All right. I feel pain or heat, okay? And instantaneously, those receptors in my hand send a signal to my brain, and my brain processes that I have touched something hot. My brain then sends a signal to an effector, which is going to be the muscles in my arm, telling my arm to move my hand away from the hot heat. Okay. This is a process that's going on within milliseconds, right? I touch the hot stove, I feel it, it sends a signal to my brain. My brain sends a signal to my effector, my muscles in my arm, to move my hand away. And I respond, okay, to that stimulus. This is happening all the time, every day. I just gave an example kind of an external stimulus, okay? But there are many examples internally where your organs are communicating with your brain, okay, through receptors throughout the body, and your brain is making changes by sending signals to effectors. So for our nervous system, we do organize it into two different sections. Okay, we have the central nervous system, which will now be called CNS throughout the lecture. And we have the peripheral nervous system, also called your PNS. The central nervous system is the brain and spinal cord. So CNS is the brain and spinal cord, and your PNS are all of your nerves and ganglia. Ganglia are just groups of nerves. And when we kind of take a look at this, you could also classify the PNS as cranial nerves, nerves branching from the brain, and spinal nerves, nerves branching from the spine. Okay, so we can kind of see that distinction here. We have the brain and the spinal cord in the center here. That's the central nervous system. And then all of that yellow, all those nerves branching off of each of those structures is part of our peripheral nervous system. We can separate out, okay, our nervous system into additional divisions. So we have central and peripheral, but we can also separate those out into sensory versus motor. Okay. And this will be a little bit different. We'll start with the sensory one. Sensory nervous system is also termed the afferent nervous system, afferent with an A, meaning, and this term afferent means that it's bringing something towards or back towards, okay? Meaning that the sensory nervous system is going to receive sensory information from the receptors and bring it back towards the CNS. Okay, so when we saw that previous kind of chart where we had receptors sending signals to the control center, this is what we're looking at here. The sensory nervous system is comprised of receptors that send signals to the CNS, which is the brain and spinal cord. We can separate those into additional categories. If these are conscious signals, we call them the somatic sensory system. If these are visceral, okay, coming from organs where we don't really perceive this is subconscious signaling, then we would call it the visceral sensory system. Somatic is something we detect. Visceral is something that we don't really perceive. It's subconscious, sending signals from internal organs. You can kind of see the examples here, okay, for sensory. The other organization is the motor nervous system, which we would term efferent with an E, and that means that it is sending signals away. And so when the brain or spinal cord processes the information coming in from the sensory nervous system, it is going to use motor neurons, okay, the motor nervous system, to send signals away from the CNS, away from the brain and spinal cord, out to muscles or glands. If these are conscious, sending it out specifically because we know skeletal muscle is voluntary, we call this the somatic motor nervous system, voluntary signals to skeletal muscles. If it is going to internal organs or glands, then we would call it the autonomic motor system or the visceral motor system, where we are sending signals to internal organs or glands. These have their own divisions, which are called sympathetic and parasympathetic, which we'll get a little more into in a later chapter. All right, so we're going to start with the microanatomy. Part 1 is just the general characteristics of neurons or nerve cells. So the neuron is our structural unit for the nervous system. This is the nerve cell. They have many characteristics, and some of these characteristics you might notice they share with muscle cells. Neurons are excitable, which means they can respond to stimuli, and there is going to be a change in their cell membrane potential, okay, meaning that there's an electrical change going on. They have conductivity, so they are able to propagate electrical signals across their cell body, which can open a number of different voltage-gated ion channels. They have the ability to do secretion. We saw in the previous chapter with muscle that neurons contain synaptic vesicles containing neurotransmitters, which they can secrete to do chemical messaging. Neurons, interestingly enough, they have extreme longevity, okay? And that kind of goes along with this last characteristic of being amitotic. Once you have a mature neuron, okay, your nerve cells, they live throughout your lifetime, okay? And if they are damaged in any way to where that neuron dies, they're not able to replace themselves. This becomes really important when we think about brain injuries or spinal injuries, or we think about even something like Alzheimer's disease, where if we lose some of these cells that have been present with extreme longevity throughout our lifetime, they're not able to reproduce themselves. So we end up losing those neurons forever because they're amitotic. So for parts of the neuron, okay, we're going to have 3 main parts, and then we'll have some separate things kind of in within those. This is a typical motor neuron. This is what that looks like. We'll look at some different shapes of neurons as well, okay, but this is a typical motor neuron, a multipolar neuron. This is its picture under the microscope. This structure right here is the cell body, which is called the soma. And it has its own plasma membrane, which we call the percaryon. Very different naming, but we've learned about some different names before with muscle, right? Where we learned that the cell membrane for a muscle cell is sarcolemma. For the cell body, the soma of the neuron, its cell membrane is called the percaryon. And it contains largely all the different structures you would expect. It has the nucleus, it has mitochondria, lots of other organelles that we would expect. But it also has these structures called Nissl bodies. And these Nissl bodies are just kind of clusters of ribosomes. And we know that ribosomes are producing proteins, okay? And protein production is very important inside the neuron because it needs to make neurotransmitters. All these branches that are coming off of the soma of the neuron, as you can see here, lots of branches, those are called dendrites. And these dendrites, we'll get into what unmyelinated means soon, okay? But they're unmyelinated and they're just branches that come off of the neuron and they essentially act as receptors, right? They receive input and transfer it to the cell body. So these branches, all they can do is receive information, stimuli, and then they send those signals to the cell body for processing. So those are dendrites. The word, that prefix or that root word dendro means tree. Okay, so dendrites kind of look like tree branches coming off of the cell body. The last structure here, which I kind of highlighted larger, is the axon. We've learned that word before because we learned about these synaptic knobs at the end of the axon, which is how our motor neurons communicated with our muscle fibers. And axons, as you can see, have these kind of structures sitting on top of them that are kind of white or translucent. And this is myelin, okay? A specific cell here is making the myelin, but we would call the axon portion of the neuron myelinated. So it has these sheets of myelin being produced by this specific cell, which we'll learn about in a minute. There are a few different parts to the axon. At the top of the axon, right next to the body, we have the axon hillock. That area is going to receive all kinds of different electrical signals that are being processed by the cell body, and they all kind of sit and come together here at the axon hillock in a process called summation, which we'll get to. And then at the axon hillock, it's going to send electrical signals down the axon. It's important to note that the axon is how the neuron communicates with other cells. So it is going to send signals through the axon to communicate either to other neurons or to muscles or glands. So we have the axon hillock here. You can see that there are some branches kind of coming off of the axon. It's not one straight line. Those branches are called collaterals. Once we kind of get to the end of our axon, okay, where we start to get to those synaptic knobs, each of these little tiny branches that we have, we actually call telodendria. Okay, telodendria have little synaptic knobs on the ends of them. And we know inside those synaptic knobs, we have synaptic vesicles containing neurotransmitter. So some of these are old terms that we learned before, right, when we were looking at muscle cells, and then there's a few thrown in new terms regarding the axon. We're going to step away from neurons real quick, and we're going to talk about nerves. And first, we're going to talk about the structural classification for nerves. So if you remember for the PNS, this is all the nerves branching off of the brain or spinal cord. If they branch from the brain, they're called cranial nerves. And if they branch from the spine, they're called spinal nerves. These nerves can follow very similar classifications to what we just looked at for neurons, okay? We have sensory nerves, which contain sensory neurons sending signals to the CNS, right? We would call these afferent nerves, sending signals to the brain and spinal cord. We have motor nerves, which are made of motor neurons, sending signals away from the CNS, efferent. And then, especially when we get into the category of spinal nerves, they share a kind of mixed function where they have both a sensory part and a motor part, meaning they contain both sensory and motor neurons. If we have a cluster of somas or neuron cell bodies making up these nerves, right? Because there's a bunch of neurons in each of these nerves, then we would call it a ganglion for the PNS. So the PNS contains our nerves, and we would call it a ganglion if we have kind of a cluster of the cell bodies for those nerves. Looking at a nerve itself, okay, and this might look very, very similar to when we were looking at the larynx for the muscle cells. A nerve is essentially a bundle of axons. Okay. We might have, and I'll kind of explain this, if we were go back real quick to the structure of a neuron. Okay, say this is a motor neuron. Okay, we see the cell body and we see the axons. When we put nerves together, okay, we've got the whole neuron and we've got a bunch of them. And if all the cell bodies of those neurons come together, they form a ganglion. Okay, but we need to see what we do with the rest of that nerve. So when we look at here, the nerve itself is a bundle of axons, okay? So if we look at the spinal cord for this example, you can see there's kind of a lump or a kind of structure here, if you can see my mouse, okay? And you can see inside, if we look microscopically, we see a bunch of the cell bodies of these neurons, and clustered together, we call them a ganglion. That ganglion has all those axons coming off of it. Those axons are going to come together to form a nerve. And those axons are going to be wrapped around by a bunch of different layers. The entire nerve itself, which is a bunch of bundles of axons, is surrounded by epineurium. Epi meaning outside, neurium, okay, meaning for the nerve. Each of those axons are put into their own bundles, okay, which we've learned this term before for muscles. And those bundles are called fascicles. And each fascicle is going to be surrounded by perineurium. Lastly, each individual axon in that bundle is going to be surrounded by its own connective tissue layer, which is called endoneurium, the most internal. And then lastly, for these nerves, aside from seeing all those different structures, you can actually see the blood vessels here in this picture, meaning that nerves, just like muscle cells, are vascularized, meaning they have a decent blood supply, okay, to keep these cells supplied with nutrients and offloading of waste. We're going to get into some structural classifications now for our neurons, okay? What we've looked at largely before right now is just, this is a typical structure of the neuron with those parts, cell body, dendrites, and axon. When those come together to form nerves, we learned a term ganglion for when those cell bodies all come together. And then we have what happens and what is wrapping around each axon, okay? Axon bundle or bundles of axons, okay, next to each other. Neurons themselves, aside from having that structure like we saw initially, do show themselves in different ways. So the one that we looked at first, and I'm just going to go back on that slide, this one right here, this is considered a multipolar neuron, meaning it has multiple poles. We can see an axon, and then we can see a bunch of dendrites coming off of it, multipolar, okay? So that's what we see with those multipolar neurons. A lot of dendrites, one axon. This is the most common type that you're going to see in the body. We have some exceptions to this in different parts of the body where we have a different shape, a different structural classification for neurons. One is a bipolar neuron, one dendrite, one axon. Places you would see this are going to be in our special sense areas. Our special senses are our sense of vision, smell, hearing, taste, that type of thing. And so an example of this could be in the retina of the eye where we see we have these bipolar neurons. And I'm going to see if I can draw real quick for you. A bipolar neuron essentially would look like this. You have the cell body, and then you have one side where we have the dendrite, and the other side you have the axon. Very different from the multipolar where you've got the cell body, you've got one axon, but then you've got a ton of these dendrites coming off of the cell, okay? So this bipolar example we see in the eye, okay? Or we see in other places where we have special senses like taste or smell. This one is fairly rare, okay? It's only found in certain parts, okay? This last classification is unipolar neurons, also called pseudo-unipolar. okay? One process extends from the cell body. For typical unipolar neurons, we typically see these in insects where it looks like this. And this again is just the structure of the neuron. Unipolar, one thing going off. And we typically see this in insects. In humans, we would actually call these unipolar neurons pseudo-unipolar because they look a little different. They don't look like insect ones. They look like this, where we have one pole, but it divides into dendrite and axon. So this is what pseudo-unipolar looks like, and this is what it looks like in the human body. So one process, we have the peripheral process, which splits into these different dendrites, okay? And then we have the central process, which is that axon that eventually has a synaptic knob on the end, communicating with the CNS, the brain. So again, typically we see these unipolar neurons with certain types of senses that we have in the body. So sensory neurons, bipolar neurons are also sensory neurons, but they're special sense ones for those special ones in the head, like taste, vision, hearing, that type of thing. And then the rest of the neurons in the body are typically look like this nice one, multipolar. That's just for our classifications here. All right. For functional classifications, we've already gotten into this a little bit, okay? We have the sensory versus motor neurons, which we would term afferent versus efferent. Afferent means these sensory neurons are sending signals to the CNS, okay? And a lot of times you see those as unipolar, right? Those sensory neurons. Sometimes bipolar for special senses. And then all the motor neurons in the body, those efferent ones that send signals away to muscles and glands, to effectors, those ones are all considered multipolar. There is a category of neurons that we haven't talked about yet called interneurons or association neurons. These ones typically receive, process, and integrate information from other neurons. And they help overall with just communication between sensory versus motor neurons. And these are all generally multipolar. I'm going to see if there's a picture on the next slide. Yeah, there is. Okay, so this picture will help that a little bit. Okay, so for our sensory neurons, we receive sensory inputs. Okay, this is part of our sensory neuron. If there's a bunch of them together, we call it a ganglia. This is the spinal cord. Sensory neuron is going to bring information to the CNS, which is the spinal cord. And then a lot of times what we see in the CNS is these interneurons, which connect sensory neuron to motor neuron. And some of these interneurons can be very long or form long chains so that they make sure that we connect one to the other. So this interneuron is connecting the signaling between the sensory neuron and the motor neuron. And the motor neuron is sending signals away from the spinal cord, from the CNS to a effector, which in this case is a skeletal muscle. So we're going to talk a bit about communication now between neurons or even between neurons and their effectors. I'll draw this real quick to kind of give an idea here. So if we have a neuron, and I'm sorry that these pictures suck, okay, but if we have a neuron here, and what I've largely drawn, as you can tell, very poorly, is a multipolar neuron, okay? And how it communicates, either we could do a muscle cell if we want to, but I'm just going to draw another neuron, okay? So I have two multipolar neurons, and we know that this is the axon right here, that single extension. And that is how all neurons communicate with other things through the axon. So we can see one neuron is using its axon to communicate with the dendrites on the cell body of this other axon. This communication point here is called a synapse, okay? Place where a neuron connects to another neuron or an effector. There's two different types of synapses, and this just depends on whether a neurotransmitter is being produced or if we're just exchanging electrical signals. Chemical ones are far more common than electrical ones. And by chemical, I mean the release of a neurotransmitter. So, say this had a synaptic knob on the end of it and it was releasing neurotransmitter, and that's how it was communicating with the other neuron. That would be a chemical synapse where we release a chemical, which is our neurotransmitter, to communicate with the other cell. There is such a thing as not releasing a neurotransmitter, and this is an electrical synapse where these neurons are bound together by gap junctions, cellular junctions, and there is just a passing of that electrical signal through each other. This is very, very fast. There's no delay. You can tell if we were to do a chemical synapse, which is most common, there's a little bit of a delay because that neurotransmitter has to go across the synaptic cleft and bind to receptors on the other cell. So that's a little bit of a slower process. Not terribly slow, but definitely electrical synapses are faster because we don't have to have that step of that chemical synapse. All right, so let's clear these drawings. The chemical synapse, which we've already looked at before with muscle cells, is a little bit different. So we have what we would term a presynaptic neuron, which is sending the signal. It's the one producing the neurotransmitters, presynaptic before. And this is before the synapse takes place, before we have that connection. And then we have the postsynaptic neuron, which receives the signal. So if you remember my picture before where I had one communicating with the other, the one that's producing neurotransmitters is the presynaptic neuron. The one receiving the signal is the postsynaptic neuron. We know that those neurotransmitters travel across a space called the synaptic cleft, which we learned with muscle cells. And this is just kind of the events of this, right? The neurotransmitter releases from the synaptic vesicles in the synaptic knob into the cleft. It diffuses across the cleft and finds these postsynaptic receptors. It binds to those receptors and creates what we would call an electrical signal. In this case, we're just going to call it a graded potential. Okay. A graded potential is when we're building up to form an action potential. And again, there is such a thing as called synaptic delay, where it does take time for this to happen, which is why chemical synapses are take a little bit longer than electrical ones. So for neurotransmitters themselves, we've only have to learn one in this class, which you've already learned in the muscle chapter, which is acetylcholine, ACH. But let's just learn about some characteristics of neurotransmitters. They are made by neurons. That's not surprising. Okay. And they're placed into vesicles, which travel down the axon into the synaptic knobs. They are released when action potentials trigger. Okay. So we have, when we have action potential triggers like calcium entry into the synaptic knob, remember when we were learning about the muscle contraction and what happens at the neuromuscular junction, in order for the synaptic vesicles to release their neurotransmitter, calcium has to enter into the synaptic knob. This is creating an action potential. All right. And we know that that calcium binds to the synaptic vesicles. And that's what's going to actually release the neurotransmitter. Neurotransmitter goes to a target cell, another cell, attaches to those receptors, and triggers a response in the target cell. And as far as neurotransmitters go, there are 100 different types, and they are classified into different groups. Where I think what I kept for this class is we're just going to call them excitatory and inhibitory for those groupings. These are the main classes for these. And I did put a note here. You do not need to memorize these. Okay, so don't worry about memorizing these. Acetylcholine is the only one that you would need to know because that's what we memorized for the muscle contraction. Okay. We are going to get into some classifications for these neurotransmitters as to whether they're excitatory, we're going to do an action potential, or inhibitory or not. But these are just a few other classes. Feel free to read through these if you like, but again, you will not be tested on these for this class. All right, here we are. So for the classification by function for the neurotransmitters, we are going to look at them as excitatory or inhibitory, okay? Um, and excitatory, again, we could call this EPSPs, and we'll kind of get into that abbreviation in a minute. I don't think I took it away. Oh, I might have. Okay. Um, this is kind of for this one, the EPSP. This is for an overall excitatory effect, causing our, neuron to have more positive signals. Okay. And remember, if we have a resting membrane potential, we can go with the example for a muscle cell of that negative 90 millivolts. Okay. Us getting excitatory signals to try to get to an overall excitatory potential of maybe net positive to do an action potential. This is what we're trying to create, excitatory actions with those neurotransmitters. This is for membrane potential. Or on the flip side, if we do not want to create an action potential, we can have an overall inhibitory effect of certain neurotransmitters, and this would be that inhibitory potential. Um, some neurotransmitters immediately create an effect. Okay, so this would be direct transmitters bind to receptors that are chemically gated, immediately creating that potential. Or they could be indirect. Some of the neurotransmitters bind, but they just start kind of downstream effects where we might involve certain things inside the cell, like G proteins and second messengers, which will have different effects inside the cell. Indirect ones, meaning that when that neurotransmitter binds, it's not necessarily going to change the electrical potential of the cell itself. We might have a few other things that have to occur inside the cell before we actually create a change in that synaptic potential. There are other things other than neurotransmitters, like neuromodulators. These are different chemicals that may act similar to neurotransmitters and alter the responses of the neurons. Two ways that they can do this is by facilitation or inhibition. So some neuromodulators actually create a greater response to a neurotransmitter. Um, and by the neuron. They do this by facilitating it. So they can essentially increase the amount of neurotransmitter in the cleft or the number of receptors on the postsynaptic neuron. So neuromodulators might do this so that we can kind of create a greater signal from that neurotransmitter. On the flip side, for inhibition, you might have certain neuromodulators that help create a weaker response, and they can do this by decreasing the amount of neurotransmitter present or by decreasing the amount of receptors on the postsynaptic neuron. We're going to pause here, and I'll have another video for part 2 where we get into neuroglia, which are the cells that help neurons survive and also help keep them in place inside the CNS and PNS.